Okay, so let's discuss about the translocation in detail, where the food, amino acids, nutrients that are transported from the source cell to the sink cell. Source is where the sucrose has been produced, the food which has been produced, and it travels down to the phloem, right, how it's shown here, and it enters to the sink cell or to the store organ, where the sucrose will be used. So let's go stepwise how this particular process occurs. So it actually occurs through the mass flow hypothesis. There were several theories which has been proposed to explain the mechanism of phloem translocation, which are this. So there were many, but the well accepted theory was Munch Hypothesis. Munch basically has explained how the translocation occurs in the phloem. He was the one who has initiated this process, and later on, he was the one who has proposed first time in 1930, and later on it was elaborated by Kraft in 1930. So let's discuss the mass flow hypothesis. Now, as in xylem vessel, the difference in pressure that causes the mass flow is produced by what potential gradient between the soil and the air, requiring no energy input from the plant, but that's not the case with the phloem transport. It's quite different. To create pressure difference needed for the mass flow in the phloem, the plant has to use the energy. And therefore, phloem is an active process, in contrast with the xylem, which is the passive process. So there needs to be an active loading of something. So the pressure difference is produced by active loading of sucrose into the sieve element at the place from which sucrose is to be transferred. It's a key terminology, active loading of sucrose. That's the first step towards the translocation. Okay, so to build up the pressure difference, active loading is required, and with that pressure difference, the mass flow of sucrose will take place, which was proposed by the Munch. So first step is active loading of sucrose into the sieve unit. So let's start with the first step, which is active loading of sucrose into the phloem sieve tubes. So I'll explain you through this picture stepwise, just view the presentation. So right here, over here, we have the leaf cell, which is the mesophyll cell, where the sucrose is produced. This is the companion cell. So let me just write it down. This is the leaf cell, which is a source. Right next to it, we have the companion cell. And this is the phloem sieve element. Now here, by the process of the photosynthesis, the glucose has been made and glucose has been converted into sucrose, where the sucrose is the one which is used for the transportation. So now what happens is, there is no initial pull for the sucrose to move this way. Sucrose is produced, it's ready, they needs to go down towards the sink. But like xylem vessel, there is no initial pull. There is no transpiration pull, there is no water difference still here. So now what is done is, active loading needs to be done over here. Energy needs to be given to take this sucrose towards the phloem. How it is done, we'll see. On the cell membrane of the companion cell, there is a presence of H plus proton pair. Here. which is the H plus proton pump. This do what is, they will allow the H plus to move into the leaf cell. Now as the H plus moves here, the concentration of H plus increases over here. So H plus will do what? They needs to go back to its original position. Of course, the ions, they will do what? They needs to go down its concentration gradient. Okay. So first of all here, ATP was used. Why? Because against the concentration gradient, the H plus was allowed to move into the leaves. Active loading is done right here. Active loading means H plus were moved against the concentration gradient using ATP, increase the concentration there for the H plus. Now since the gradient is formed, since the concentration of H plus has increased there, H plus again needs to come back. So they will come back through a co-transporters which are present on the cell membrane of the companion cell. These are the co-transporters. The role of the co-transporter is, along with the H plus, it will also take sucrose to the companion cell. Okay, co-transporter molecules, they are the carrier proteins. They will do what? They will take the H plus, who is already concentrated in the leaf cell. Along with the H plus, they will also take the sucrose to the companion cell. Now here our aim is achieved. We have the sucrose at our companion cell. Okay, so let's just quickly recap what was our aim. Sucrose has been produced in the leaf cell. Now there was no initial pull. Nothing was there to push this sucrose down to the companion cell. There needs to be some initiative energy to be given. For that, there are H plus proton pumps present on the cell membrane of the companion cell, which has allowed H plus to move to the leaf cell where the concentration was low. So it's against the concentration gradient. It is moving from high to low. Therefore, ATP was required. That's the H plus proton pump. Now, H plus, since the concentration has increased, they will move down again to the companion cell by the co-transporter protein, which are carrier protein. So, along with the H plus, they will not come alone. They will also take the sucrose down to the companion cell. And our first step is done. Loading of sucrose has been successfully done into the companion cell. So here the ATP is required. That's why it's an active loading. Now this sucrose can easily be moved down into the sieve plate through the plasmodesmata which are present over here. Sucrose will move down. And now sucrose is present here. They have traveled down through the plasmodesmata. So now sucrose is present right here in the phloem seal. So you can quickly just see through this particular photograph right here which I have. Loading into the phloem. This is the source cell, companion cell. How it is done is here. There were first of all proton pump present in the companion cell membrane of the companion cell. It has allowed H plus to go there. Against the concentration gradient, therefore ATP hydrolysis has been taken place. ATP has hydrolyzed and released one phosphate. One phosphate bond has been broken up, which has provided energy for this pump to allow the H plus to move into the leaf cell. Now, H plus will come down to the co-transporter. Co-transporter has a tendency to always take two passengers with them. Okay. Then, therefore, H plus along with the sucrose can now easily enter the companion cell. Once in the companion cell, the sucrose has come. Now, the sucrose can go down into the phloem tube through the plasma vesicle. So, this is the first step. Active loading of the sucrose into the companion cell and then into the phloem tube. Now see, just a bit of extra knowledge I would like to give here is, in some plants, there will be symplastic phloem loading, where a symplastic loading means where sugar moves from companion cell into the sieve element through the plasmodesmata itself. But when there is apoplastic pathway, where ATP is also required and sugar moves through the cytoplasm. So whenever the sugar is moving through the cytoplasm, apoplastic pathway is taking place. At that time, this loading needs to be taken place. Okay, so if it is taking symplastic pathway, easily it can move down through the plasmodesmata. But when it is moving down through the cell wall, apoplastic pathway, it needs to be done through the active load. There is the requirement of H plus proton pump. Generally, apoplastic phloem loading is seen in most of the plants. Now, first step is achieved. That is loading of sucrose into phloem. Now, next step is mass flow hypothesis. Where now the phloem will travel down because of the hydrostatic pressure which has been created. The mass flow hypothesis which has been given by Munch. So the loading of the sucrose main aim was to increase the concentration of sucrose into the phloem tube. Now what happens is in the mass flow hypothesis. Now before going into the mass flow hypothesis, I would like to just give you a short introduction using this model. That the source cell, it is photosynthesizing cell, so there the sucrose concentration is high. So since the sucrose is high over there, of course the water will be low. So water potential will be low inside the mesophyll cell. So what happens is the water outside where the tank water is present, it will start entering inside the sucrose. That's the first scenario. Why? Because source cell is producing more of the sucrose, so therefore there is more solute concentration. More solute concentration leads to less water potential. Therefore, the water will start entering inside the source. On the other hand, into the sink cell, what happens is, sink cell, since it is using those sucrose, it is allowing the sucrose to reduce its concentration. Therefore, solute concentration is low here. And if solute concentration is low, water potential will be high. More water potential there, so water will start losing out from the sink cell by the process of osmosis. So this has created a hydrostatic pressure. So over here you have the increase in hydrostatic pressure where the water is entering inside the source cell, and here there is decrease in the pressure in the respiring cell. Why? Because the sink cells are using more of the oxygen. Because of this difference only now there will be the movement of the source, sucrose. So the source cell has the higher hydrostatic pressure than the sink cell, so the solution is forced towards the sink cell via the fluid. This is just a simple explanation to make it simple concept. Now I'll teach you in depth. But I want you guys to understand the concept of hydrostatic pressure, that why there is high hydrostatic pressure in the source cell and why there is low hydrostatic pressure in the sink cell. So now taking that particular diagram in mind, just see over here that in the sieved elements, now there is more solutes. Solute concentration is high. Solutes are high. Water potential is low. So what happens is, this right here is the xylem vessel. Of course, the xylem and the phloem will be running parallel to each other. So now what happens is, the water which is running in the xylem, it will be entering into the phloem sieve element by the process of osmosis. This is happening because of the difference in the water potential. Here the solutes has increased, therefore the water potential will be low. And here the water potential is high. So water will move from high hydrostatic pressure to a low hydrostatic pressure. Okay, so that's what is taking place. Water uptake will take place by the osmosis. So the loading of solutes, that is sucrose, from source cell into the phloem sieve tube has increased the solute concentration and decreased the water concentration. Because of this, the sieve tubes has to take up the water by the osmosis, or the xylem is right next to it, so they will take the water through the xylem. So there is raised in hydrostatic pressure, which will causes the content of the phloem to flow towards the sink. So that's what the pressure flow hypothesis is. Because of the loading, there was the high concentration of water here. Hydrostatic pressure has increased. The sieve tube cell wall resists the deformation caused by the water movement down the concentration gradient into the tubes. That creates the positive hydrostatic pressure here. Forces content of the phloem sieve move along the sieve tube in the bulk flow. Why? Because the pressure has created over here. Here there is already low pressure. So the gradient of the pressure in the sieve tube is maintained by the active unloading of the sucrose at the sink. Now since it has reached down because of the difference in the pressure, okay, there was high hydrostatic pressure here and low over here. Now to maintain this pressure, unloading will be taking place into the sink, that is the root sink. Right here. There also requires an active unloading. And the loss of the water by the osmosis will take place. So because of the incompressibility of water, it allows transport along the hydrostatic pressure gradient. That is the overall scenario, which is right here, creation of hydrostatic pressure, how it has been created. I hope you are now well understanding that because of the loading of the solutes, there was the creation of the more hydrostatic pressure here. As the water potential was getting low here, the water has traveled from xylem into the flow. High hydrostatic pressure has been created, and because of that, the water has moved down. Along with the water, the sucrose has moved down. Now to maintain this pressure, there will be unloading taking place in the root cell. Now we want to unload the phloem, which was we want to unload the sucrose, which was carried out through this phloem. So unloading of the solutes, that is sucrose at the sink, and loss of water back to the xylem will create a low hydrostatic pressure there. So hydrostatic pressure gradient from high to low moves sucrose and other solutes to the sieve tube from the source to sink. So because of the hydrostatic pressure built up here, the pressure created because of water, here it was high. Because of that, the movement of the solutes along with the water has moved down. solutes over here, I mean to say, is sucrose. It will move down to the low hydrostatic pressure, and here again the unloading will take place. Companion cell we have, and we have the root cell. So this is the root, and this right here is the companion cell. So again unloading will take place. Unloading of what? Unloading of sucrose. Again the same procedure takes place, and here the sucrose will be present. Now the sucrose can be either used for the respiration or it can be stored. So that's what takes place at the sink. From the sieve tube to the companion cell, the solutes, that is sucrose, has been reached. With the help of the ATP, maybe it will be again used basically in the fruit, which has been converted into sugar, or it can be stored. So that depends on the cell. So that's how whole of this process takes place. Photosynthesis taking place in the source cell, which is leaf. Carbon dioxide plus water, sucrose is made. Sucrose loading is taking place into the sieve tube through the companion cell. Here then mass flow is taking place because of the pressure difference, hydrostatic pressure. Unloading is taking place into this growing or the storage point. Now here water and sucrose may be combined and give the glucose fructose. Either by the respiration, it will go for the process of respiration, or it will go for the storage, or it will go for the... So this is the overall scenario which is taking place. Right here, the loading requires the ATP. And that loading is necessary to create the pressure difference, hydrostatic pressure difference, and because of that the mass flow is able to take place. So I hope now you guys are clear with the translocation. Active loading of phloem and the mass flow. Active loading is very necessary to create the difference in the pressure. Now there is one experiment which is done to prove that organic solutes are translocated through phloem only and not through the xylem. So to determine whether the xylem or the phloem tissue is involved in the translocation, it is possible to remove the cortex and the phloem of the stem in the form of the griddle or in the form of this ring. So what happens is, when you remove this part right here, you will observe that the tissue above the ring has a swollen region with a sugar accumulated down there. So from here now, no transportation of organic solute has taken place. Therefore, you will observe this region, tissue below the ring, they begin to die since no sugar has been transported across the ring. So here, first of all, what they have done is they have only removed the phloem to check that whether phloem is present in transportation of organic solutes or not. So they have just removed the phloem and xylem was enough. What they have observed is here there was bulge. Swollen has been created because sugar accumulation has taken place here. And down the part, the below tissue, there was completely damaged. So therefore they began to die. No organic solute has transported. This proves that phloem is the one which is responsible for translocation process.